Hi friends, my name is Partha Varanashi, and we also have Roshan Shetty right behind the camera. On behalf of Varanashi Research and Development Foundation and Discover Agriculture, today we're going to talk about soil microbiology and its importance in the regenerative farming. For a long period of time, to be exact 450 millions of years, we have seen the regenerative cycle that happens in the nature. So we see it in the forest the most, wherein the carbon cycle or the nutrient cycle happens due to the presence of all various types of organisms that are present in the nature. So the plants produce your fruits, and they are eaten up by macrofauna such as animals, and then they excrete. That excretion is further eaten up by various kinds of soil microorganisms, and then they finally break down this organic matter back into the soil as nutrients in the form of nitrogen, phosphorus, potash, micronutrients and secondary nutrients, which are absorbed by the roots, which goes back up again for photosynthesis, along with the presence of sunlight, carbon dioxide, oxygen and water. And then again we have the fruit, and then that falls down again, eaten up by animals, and then the whole process repeats. Now in the forest, this process is really fast because of the presence of all the different components of the nature which are present there. They are living organisms, the macroanimals, the microfauna, and of course the soil biology. Now when we talk about soil biology, there are 5 different types. There are the bacteria, there are the actinomycetes, there are the fungi, there are nematodes, and also, what did I miss? There is I think yeast is another one. So there's 5 different types of soil-borne microorganisms, and their presence is essential for this nutrient cycle to happen. Now we all know that we have seen Sadhguru from India traveling around the world talking about the life, the health of soil. He's on this mission to save the soil, so he's talking nothing but how we are losing this soil microbiology. The species, about 27,000 different species of soil microbiology we are losing per year on the planet. As this is happening due to various reasons. One is excess use of chemical fertilizers, the other one is doing monoculture, and basically creating an atmosphere which is not suitable for the flourishment of microorganisms. So now let's talk about how we can improve the microorganisms. So these microorganisms which basically degrade the organic matter back into the soil. As we can see, the organic content in the soil is so high. This is because of presence of all the different organisms here. So how what we can do, like what we have done at our farm for the last 30 years with the help of our research center in Varanashi Development and Research Foundation, we have isolated certain microorganisms which are highly beneficial for the plants. So there are the nitrogen fixers, there are the phosphorus solubilizers, there are the potash mobilizing bacteria, and there are biocontrol agents, and there are microorganisms which produces, like bacteria which produces plant growth hormones, and they produce the secondary metabolites which improves the immune system of the plant. So there are these various organisms that they are present in the soil which are championing the symbiosis between plants and the soil. So in their presence, they keep the bad pathogens away. They help the degradation of nutrients and make it bioavailable for the plants. Giving you an example, the things like nitrogen needs to be fixed into the soil into bioavailable form. Phosphorus can easily react with cations such as iron, magnesium, zinc, and form complexes which are not available for the plants to be absorbed. So these microbes, they release certain enzymes which break down these complex molecules into simple form of nitrogen, phosphorus and potash, and also the microbes and the secondary nutrients which are readily absorbed by the plants. So this is why the presence of microorganisms is very, very essential in our farm. Just by putting chemical fertilizers, they're just going to be around in a complex form, but they're not available for the plants. So it's very essential that we inoculate these biofertilizers or beneficial microorganisms of various kinds. There is a vesicular or Arbuscular mycorrhiza, there is the effective microorganisms, there are consortium of different species of nitrogen fixers, phosphorus solubilizers, potash mobilizers. We will talk about it in this video. We'll take you to our microbiology lab and we'll show these different species that we have gathered in the last 3 decades, what we've been using in Varanashi farms. And this is how one of the primary reasons, along with there are other reasons like multi-crop system and the permaculture practices, but one of the main reasons we have this regenerative system at Varanashi farms is because of the presence of beneficial microorganisms, or what we call as biofertilizers and biocontrol agents. Come, let's go to the lab and have a look at these amazing species of microorganisms which gives us these benefits. Come, let's see our soil microbial mother culture bank. So this is where we have kept all our the different beneficial microbes in a petri dish, and we keep subculturing it every week. And let's see which all we have here. So in here we see Azospirillum. We have Pseudomonas. And we have Rhizobium here. It's been inoculated on the 24th of February. And here Bacillus Subtilis. We have Bacillus Circulans in here. And Bacillus Megaterium. Frateuria, which is a potash mobilizer. A nitrogen fixer, Azotobacter, inoculated on the 24th Feb as well. And then we have some fungal mycelia. This is Trichoderma. You can see this we recently obtained from a university. This one's something that we have maintained here for the last 15 years. Again, it's been subcultured every week. And here we have another fungi, which is Aspergillus Niger. Hello friends, in front of me I have various biofertilizers, the soil-borne microorganisms which are beneficial for the plants. They do two different things. They protect the plants from biotic and abiotic stress. So biotic is basically an infection caused by another microorganisms or another living being, as it says. And abiotic stress are the ones which is caused by drought or higher salinity or higher pH, all these different situations of the environment. So I'll take you through certain microorganisms which are in front of me, which have been isolated and maintained at Varanashi Development and Research Foundation, and we also produce this through Varanashi organic manures and sell it to the farmers so that they can enrich their land with these microbes. So first, let's start off with Azotobacter. Now, Azotobacter is a plant growth promoting bacteria. It is found in soil, obviously. So once inoculated into in this carrier medium is coffee husk, we spread it across the soil in our plantation. So what it does is it's a nitrogen fixing bacteria. It fixes the atmospheric nitrogen into the soil, makes it bioavailable for the plants, and it also produces certain phytohormones which reduces the effect of biotic and abiotic stress that's experienced by the plant. So that there we have Azotobacter. It's also known to produce indole acetic acid, which is a growth hormone, and it's basically overall the presence of Azotobacter in the soil enhances the plant production. It keeps the pathogens away, and also it fixes the atmospheric nitrogen into the soil, making it bioavailable for the plants. Alright, so next up we have this guy. This is Azospirillum. It is also a nitrogen fixing bacteria. It's a free living bacteria. It's not it's a symbiotic, and it also produces phytohormones which kind of helps the plant fights off abiotic stress, such as higher salinity, variation in pH, drought, and also the presence of heavy metals sometimes regulates the plant growth. So this helps the plants fend it off, and also it produces growth hormones, so helping the plants to grow better. So that is Azospirillum. It is a bacterium as well. And then we have let's go into Trichoderma here. Trichoderma, one of our favorite. This is a fungus, and it's also a biocontrol agent. So when there is Trichoderma growing in your farm, it keeps away the pests like the pathogens such as Fusarium and Phytophthora away. So we know that Fusarium and Phytophthora causes the root rot and the wilting and the root rot disease respectively. And this is a natural enemy of Phytophthora and Fusarium. So in the presence of Trichoderma, they do not, they don't thrive. The pathogens do not thrive in your farm. Thus, this protects your plants from these diseases. At the same time, it's known to produce indole acetic acid, which is a phytohormone which increases the shoot growth. So that's Trichoderma. You can see the colonies and spreadings over here. It's come beautifully. So at Varanashi Research Foundation, my father, Dr. Varanashi Krishnamurthy, found this accidentally. It's the first biofertilizer that we started in here. It came in as a pathogen for mushrooms, and when he was studying what it was, he read different articles, science articles, and found out that it's a biofertilizer. So this is how our story into biofertilizer started 3 decades ago. Trichoderma. So next up is Pseudomonas, which is in my hand. This is a quite famous fertilizer. What Pseudomonas does is it kind of dominates in the rhizosphere, and it keeps the pathogens away. It is a phosphorus solubilizer. It makes the phosphorus more bioavailable for the plants. It also produces phytohormones such as auxins and gibberellins, which are both shoot and growth promoters. So it acts as a growth promoter. And it also produces secondary metabolites, which keeps the pathogens away and keeps the plant healthy. So that is Pseudomonas. And Pseudomonas grows well along with certain other biofertilizers such as Aspergillus and Trichoderma and some bacteria as well. So they all group up together and give that support required by the plants. These guys support it. So there you go, Pseudomonas. And then next, let's have Aspergillus Niger. It's a fungi, and Aspergillus also produces phytohormones. It supports the plant growth. It is also a phosphorus solubilizer, just like the Pseudomonas. And all these fungi work hand to hand together and help the plants grow faster. So Aspergillus Niger is also the one you have it in my hand. It's a great fertilizer which grows in the Rhizospirillum and supports the plant growth. Alright, so next up we have a bacterium, and this is Bacillus Megaterium. Now, Bacillus Megaterium is a gram-positive, rod-shaped, endospore-forming bacterium. It's an effective soil inoculant, so once it's in the soil it multiplies rapidly and it doesn't allow the other pathogenic bacteria or fungi to come near it. So that way it is a great biocontrol agent. It's also a phosphorus mobilizing microorganism which allows the plant to get the phosphorus readily. Bacillus Megaterium is also found as an effective biological control system for the rice root knot nematode. So there was a greater than 40% reduction in nematode in the presence of Bacillus Megaterium. So it's also a fantastic biocontrol agent and also a biofertilizer. So that's Bacillus Megaterium. Okay, so next up we have in our collection Bacillus Subtilis. Now Bacillus Subtilis is a gram-positive endospore forming bacteria which is also a biocontrol agent. It's found in the soil, it's found in the root zones, it's found in aquatic environments and also just present in the soil. So Bacillus Subtilis exhibits both direct and indirect biocontrol mechanism to suppress the disease caused by pathogens. So the direct mechanism is basically it produces secondary metabolites such as hormones, cell wall degenerating enzymes, antioxidants which assist the plants to attack these pathogens and kill them off, fend them off. And the indirect mechanism includes the stimulation of plant growth and induction plant growth hormones and also induction of acquired systemic resistance. So year by year, in the presence of Bacillus subtilis, the plant identifies the pathogens and produces certain mechanism it needs to restrain these pathogens and grow happily. So that's Bacillus subtilis. Along with it, it is also a nitrogen fixing bacteria and also helps in phosphorus solubilization. So multiple benefits, and it also comes up in a consortium of other these beautiful microbes which assist the plants all together. So there you go, the universe has created Bacillus subtilis with a certain certain thinking, and I think it's beautiful. Alright, so next up we have Bacillus Circulans, and recently they did a study in Kanpur where they inoculated Bacillus Circulans into the fennel producing fields, and apparently the color and the taste of fennel seed was much brighter and tasty, crunchier after using Bacillus Circulans. Bacillus Circulans goes really well with Vesicular or Arbuscular Mycorrhiza, and in the root zones it helps solubilizing of potash and makes it more bioavailable for the plants. The problem with the phosphorus is it combines with iron, zinc, magnesium, and it is not bioavailable for the plants. Even though phosphorus is present in the soil, when you do the soil testing, it just comes up as like a phosphorus deficient. That's not because phosphorus is not present there, it is not bioavailable there, it just simply combines with other cations and forms a complex, making it not bioavailable for the plant. So that's why we need phosphorus solubilizing bacteria and fungi, not just the circulants, all the other I mentioned earlier as well, which goes and separates these phosphorus from the cations and makes it more bioavailable for the plants. So if your place is phosphorus deficient, that doesn't mean that you should put more phosphorus into it. You should probably inoculate more phosphorus solubilizing bacteria or fungi, which is going to help you improve your phosphorus or the nutrient availability for the plants. So along with this, it also has plant growth promoting hormones, phytohormones. So overall, Bacillus Circulans is also a fantastic biofertilizer. So next up is the Frateuria. Frateuria is the only potash mobilizing bacteria that we have. It is known to increase the efficiency of photosynthesis, and also the quality of the fruits is apparently improved in the presence of Frateuria, because it makes the potash more available for the plants. It mobilizes the potash for the plants. So here we have Frateuria. Next up we have in my hand is an inoculum of effective microorganisms. So basically it's a liquid consortium of photosynthetic bacteria, some yeast, and also some Bacillus species of bacteria. So this is a great degrader of organic matter. So in the presence of effective microorganisms, it goes into the soil when it's, so you can inoculate this into your ponds, irrigation ponds. If you inoculate your ponds every 2 months, so take up the inoculum, put it in a drum, like a 200-liter drum with the 4 kilos of jaggery, and mix it up, stir it up, and you inoculate it to your irrigation pond, and then you irrigate it. Once it reaches your soil, it helps in degradation of organic matter in the soil. It enhances the root growth, and also makes these nutrients more bioavailable for the plants. And also another benefit of EM in your pond is it doesn't allow the algae to grow. So the algal growth reduces, and then that will help you keep your pond water clean, and also your pipe systems clean. So that's EM. So it's also goes really well with the other consortium of biofertilizers. And then we have VAM, Vesicular or Vascular Mycorrhiza. There's one called Chlomus species, which is produced by Cosme Biotech in Goa, and we use it quite a bit. So what VAM does is it's a fungal mycelium which connects the roots from one plant to another, and also it travels widely under our plantation. So like in the forest, the VAM connects the roots, onto the roots and spread across the field, collecting the water, nutrients from all different places into your plant roots. And also another thing it does is it gathers information from various parts of the field, and it gives that information to each and every plant it's connected to, which is all the plants present in your plantation. And say if there is drought coming up, or if there is some biotic or abiotic stress like pathogens, or some disease coming up with the pathogens, or some there is lack of water, there is drought coming up, or there is higher salinity, it gives this information into all the plants, so that they can prepare themselves, so that they produce phytohormones. So the consortium of microbiota around it produces whatever is required for the plants to improve its immunity and its sustainability. So this VAM gives them that information early on, early warning system, we can call it. So that's Chlomus species VAM. Now we've used all these consortiums, so other than EM and VAM, all these consortiums are inoculated into these two packages. The carrier medium we have used is this one microbial and one fungal. The carrier medium we've used is coffee husk, sterilized coffee husk. Coffee husk not only has things like nitrogen, the nutrients required for these microbes to grow, but also gives the space for them to grow together. And packaged this together as the Varanashi Composter. You can get this from us, and all you need to put is 30 grams of it into your plant basin. We have also another consortium called Trichoplus, and Trichoplus is basically the consortium of biocontrol agents. They keep the pathogens away, and again this is also 30 grams per plant base, along with some mulch, leaves, and some water, so that they grow in that vicinity. So these biofertilizers are available in the market, and we know that every year we are losing about 27,000 species of microbiology, which is essential for the growth of the plants as they are symbiotic in nature, and we're losing them. So 27,000 species of microorganisms are lost every year. We know that Sadhguru is doing a fantastic job traveling around the world, bringing awareness on about saving the soil. So he's talking nothing but saving the soil biology, and it's important that we bring all these biofertilizers, or the soil microbes which are beneficial, back into the soil, and that's what we are doing, and I think that's what every single farmer should be doing as well.